So my name is Anton Löfgren. For those of you who don't know me, I'm doing my PhD in the chromatography group. And I would like to tell you a little bit of why I think it's so important. So there's a difference between biopharmaceutical drugs and chemically synthesized drugs. You see a sample from the bathroom cabinet. This is the cooler variant, the biopharmaceuticals. The synthesized drugs, they are smaller, quite simple, and not so complicated. So the biopharmaceuticals, they're of the very large proteins, such as insulin, cancer medicine, or specialty drugs. But to produce such a drug, it's a very long, complicated, expensive process. You have to pass through a lot of clinical stages to prove that it's not too dangerous, harmful, has many side effects. And the average cost to bring a new drug to the market is actually around $800 million. But one question is, how is it possible to offer specialty drugs to rare diseases where there's only a few patients around the world? And this is the question I want to give an answer to. So some background in the process of developing these drugs. You have the upstream, where you basically grow the drug, and you have the downstream, where you purify it from all the impurities. So it's separated on several characteristics, such as size, charge, and polarity. And the process, of course, differ greatly among different drugs. The fact is that the majority of costs in full-scale manufacturing is actually in the downstream process. So this is why we're working here, to make it more efficient, time-saving, and make it easier for research. So one way to make it more efficient is to lump everything together, all the steps, and integrate as many process steps as possible into the same unit. So this is one of the units from our lab that we have tried to stuff as many stuff as possible into it, do everything at once. And there's a chromatogram here showing that what we have done. We've produced a drug from a company that we work with. And you can see that you cycle the drug many times, from column to column, and we've done it here 3 times. And in the future, you want to add so many different stuffs and process steps as possible to this machine. So that's it. Thank you very much.
Thank you. What you're doing is both the laboratory work in your group, yes, and the mathematical modeling of what's going to happen.
Yeah.
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 Why do you need both?
I mean, it's to know what happens. You can optimize stuff in a computer that will take years to optimize if you do it for real. Yeah, it's just time-consuming and it's so inexpensive. Yeah.
Is this done because pharmaceutical companies are mostly interested in the actual compounds and are spending time developing the compounds, and actually do not have time to do the process engineering for the downstream? Or is this, do they have the competences themselves to actually do this?
This is where we come into the picture.
Yeah.
To show them that it can be done in the first place.
Yeah.
Because it's such a slow-moving market, and they don't really believe that it can be done. So this is why we at the university can show that it's possible, and that you can save a lot of money doing this, and shorten the times.
How far away do you think that you can have a production process in that scale that you actually put in a clinic to produce the pharmaceuticals on site? Would that be possible?
Yeah.
Yeah.
Yeah. Oh, absolutely. I mean, you have to grow them as well, and that takes a little time. Yeah, but yeah, it's not difficult.
Simple and easy.
Yeah.
Good. Thank you.